not folded. This one is wrong. This one is correct. So, what is the importance of coiling? Now, this coiling reduces the speed so that you give time to give enough time for absorption. It therefore means if you could eat food and then within a short while, you will go for long call. It means that food has not been given enough time to be absorbed, even if digestion took place. So that is the reason. Then from there, we still have this tube. We also have the tubular structure. This area is like a tube, like a pipe. What is the importance of this? It is narrow. There's a very narrow. Let me emphasize this. Very narrow. We don't call it tube. We call it lumen. Very narrow lumen. The reason for this is to increase contact of food and the wall. So that is another very, very important adaptation. Then still, as we look at this, we may not see it from here. But number 4, we have the inner lining. If I take this to be the inner lining, inside it has structures that look like fingers. We call them villi. So, numerous villi, finger-like structures. If I may draw one in a very simple way, it looks like that, generally. And then each of these have what here appears like just small hairs. If I would magnify this, one cell will be like that. So these are the micro villi, and this is the villi. This is the epithelial wall. Epithelial cells, because it's one. You have to be very careful with the plurals and singulars in biology. So, this villi, just the presence of the villi is not enough, because there are a number of adaptations with the villi. Number 1 is their number. They are very many. We use the term numerous. If you just say that the inner wall of the alimentary canal, the ileum, has villi, you don't score anything, because they could be 2. So they are millions and millions. So we say are numerous. Reason to increase surface area. So are numerous, they also possess villi. So when microvilli, so when you are talking of villi and microvilli, you give it as one point. They possess villi, that finger-like projection, then some other projections on that villi, microvilli. These are meant to increase surface area for absorption. Now, this villi, again, inside it has blood vessels which have not drawn there here. And they are usually very many blood capillaries. Why? This will enable quick removal of food that has been absorbed. And why do we have to remove the food very quickly, immediately the food is absorbed? This is to ensure that you retain what we call steep diffusion gradient. Because the rule for diffusion says, the bigger the difference, the better the process of diffusion. The other adaptation that we still have here, and which I have not drawn because of the size of the space there, is a system we call a lymphatic system. Here we have a lacteal which is connected to the lymphatic system. That system is for absorption of fats, which is absorbed under fatty acids and glycerol. So here we have this is where we have the system for correcting the absorbed fatty acids and the glycerol, the lacteal. Then here we have blood capillaries. These ones are for uptake of any other substance. It is only the lacteal that deal with a correction of absorbed the fatty acids and the glycerol. Otherwise, the capillaries are for the uptake of all the other materials. We don't just say that we have capillaries. We say that the villi is highly vascularized. Vascularized, which is equals to, if I may use mathematical language, large numbers of blood capillaries. So in one time in biology, when we say that a tissue, an organ is vascularized, it means it possesses very large numbers or numerous blood capillaries. And when we do that, then our wall is very well adapted for the absorption of these substances. So next now, we shall try to answer the question. Now that absorption has taken place, what next? Then we move to the next item of agenda is none other than assimilation. We shall learn in our next lesson. Otherwise, thank you very much for listening to me, and let's meet next. Thank you.